From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on November 7th, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 116th episode of Matters Microbial. Thanks for every view, listen, or comment, and please spread the good microbial word. Last week was my favorite holiday, Halloween. Please enjoy this wonderful LuxArt portrait of Edgar Allan Poe, the OG, painted with luminous bacteria onto a petri plate by my very talented spouse, Dr. Jennifer Quinn. I do my best to promote microbial awareness, microbial centrism, and positive microbial PR. Here is a glow-in-the-dark enamel pin I just commissioned showing that indeed everyone can be a micronaut. For an upcoming conference in San Antonio, Texas, Dr. Quinn and I designed these 3D printed glow-in-the-dark pins celebrating every microbial enthusiast. Hopefully you. Dr. Quinn also 3D printed a bacteriophage. I then stuffed the hollow print with small LED lights, like DNA, that can blink in response to sound. I call them discophages. Please enjoy the light show. 2 weeks ago, Dr. Jessica Mark Welch of the Forsyth Institute was on this podcast discussing human oral microbiota. Micronaut Claire, inspired, swabbed around her tongue and teeth and swabbed then onto a nutrient plate. As you can see, there are fluorescent microbes there. Amazing. It's true that we move through a sea of microbes at all times. After all, first evolved, last extinct. In the lecture portion of my Bio 350 course at the University of Puget Sound, I will sometimes challenge students to respond to a prompt. So I asked my micronauts to respond to this prompt. What does a microbe dream about? Micronaut Kaya sent me this wonderful piece of art of a microbe dreaming of growing strong, improving its genetics, and fending off antibiotics. Creativity is so important to learning objectives. On the first day of class, I gave each micronaut and each guest of this podcast a 3D printed tardidoc, a tardigrade fused with, yours truly, seen here. I asked students to send me photos of their tardigrades on their journeys through life. Now, many are too busy to do that, but when they do, it's wonderful. As you can see, this micronaut has been taking a 3D printed tardigrade to swim meets, and I hope to see many more of these. In my microbiology course-related lab, the micronauts have fun and learn a new microbial perspective, as seen here. They truly are wonderful people, and I'll miss working with them. Recently, we've been testing bacterial isolates from moss and lichen samples for antibiotic properties. Here's a control plate. The bacterium Micrococcus luteus is spread onto a nutrient plate and challenged by various candidates. As you can see, some isolates have a strong antibiotic effect visible as zones of inhibition. By the way, the strain labeled AUG was isolated from a reusable water bottle. Pretty amazing. Here's a plate showing how 3 micronaut isolated bacterial candidates perform in this assay. And as you can see, some isolates have a strong effect against Micrococcus, and others a very modest to no effect. What's important here is that the isolates are found by the students. This assures a sense of ownership and enthusiasm and student interest. I'll keep you all posted. Several times on this podcast, we have discussed the microbiology of the built environment from various perspectives. But one component of our built environment is the building material concrete, found almost anywhere people are. Are microbes involved with that material for positive or negative reasons? So it's a pleasure to welcome Dr. Julie Maresca of the SUNY College of Environmental Science and Forestry to discuss this and related issues. Welcome to the Quality Quorum today, Julie. It's truly a pleasure to meet you.
Thanks, Mark. I'm so excited to be here. This concrete project is one of my favorite projects.
Well, let's get started in the proper way. Most people have seen concrete, but few people know what it is. Would you mind? And by the way, I don't know where this expression thumbnail sketch comes from, but I like to say it a lot, apparently, or so I'm told. What is concrete and why should we care?
Yeah, so concrete is the most used building material in the world. It's really foundational, as you said, to a lot of the structures that we have in our daily lives. I put a couple of pictures in our folder that we can use here of concrete sidewalks, concrete buildings, but we also use it in bridges, we use it in dams, we use it in roads, foundations. I've seen it recently in counterweights for like a lock system in a canal. We use it everywhere. We use so much of it that we can literally see it from space. If you look at satellite images, you can see, oh yeah, go ahead.
I was going to say, it's kind of a pun to say it's foundational when you think about it.
Yes, the puns just write themselves. I've called the title for this seminar has been Hard Microbiology for almost 15 years. It's so terrible.
So seen from space.
So yeah, we can see it from space. I also put a Google Maps image of the northeast area of the United States, and you can see all the big cities. You can see Boston, you can see New York, you can see Philly and Baltimore, everything that's connected by I-95 from space. That's how much concrete we use. And what that means is that this is a really, really common habitat. And it's made of four main components: gravel, sand, cement powder, typically Portland cement, and water. And there can be a few other things in there depending on the purpose. The ratios of those components can change depending on the structure that you're building. But those are the main ones. Once it cures, we have a material that's really hard, it's really dry, it's really salty, and it has this insanely high pH. When it's freshly poured, concrete has a pH of about 12.5, which is the same as bleach. Yes, this makes me extremely popular at cocktail parties. So it's really, even though it's a very common habitat, it's a really challenging place to live. But when we started thinking about this, a colleague of mine in my previous department, which was a civil and environmental engineering department, Thomas Schumacher walked into my office and said, we should collaborate. He was a structural engineer. We should collaborate and we should make bacteria fix concrete. And I said, you're crazy. Get out of my office. We were both pre-tenure. We couldn't afford any craziness, right? But then I read some papers and he sent me some papers and I said, okay, you're not as crazy as I thought you were. But he was really focused, and I can see it. There's a lot of people thinking about this on microbes as engineers, microbes as things that can live inside concrete and fix it as it breaks down. So as problems are caused, can we fix them when they're small enough that microbes can repair those? So thinking about biorepair of concrete. I put a little cartoon of a microbe doing bricklaying because we were kind of thinking about them as masons. But there's a lot of people with expertise in biomineralization who are working on that. And it's really not my field. So we started thinking instead about concrete as an environment, because that's my background, right? I'm an environmental microbiologist. I think about which microbes live where, what are they doing in those places? And because I spent a long time in civil and environmental engineering departments, are there ways that we can put them to work? So when Thomas first proposed this to me and I told him he was crazy, I really did think he was crazy. I thought that this was too challenging of an environment for microbes to live in. But then I did start looking around me, right? Is there life in and on concrete that I can see with my eyeballs? And of course, we've all seen, grass growing next to our sidewalks. So that's pretty common. And I was like, okay, so concrete doesn't destroy everything around it, right? Those high salt, high pH conditions don't leach into the soil, or at least they don't leach it into the soil enough to make it a sterile environment. And then I visited my parents and saw zinnias growing out of this little crack in concrete in their backyard. I put a picture of that in our folder too. And we've all seen that too, right? Dandelions and other weeds growing up out of those cracks. And so again, there is some life in concrete, and some of it's big enough that we can see it. And then I started looking more closely, because I am a microbiologist, and seeing that on walls and stairs that haven't been power washed in a while, you do start seeing biofilms growing. So I put a picture of that in here too. So there are, you mentioned lichens earlier, and there are lichens that grow on concrete. There's mosses that grow on concrete, and probably those are growing on top of thin layers of bacteria underneath. So I decided that Thomas was probably not crazy, and we did start thinking about a real project looking at what kinds of microbes live in concrete. And initially, I was still pretty worried that if I put a graduate student on this project, I would be setting them up for failure. Like, there might not be enough in there for a graduate student to graduate, and I don't want to set anybody up for failure. So I had a bunch of very brave civil engineering undergraduates start this project, because they can graduate even if their projects don't work. And where we started was cultivation. Mm-hmm. So I know this is an approach you take in your classes. And basically what we did was take pieces of concrete, drop them in some buffer, vortex for a little bit, and then spread them out on different kinds of media. And we saw actually a fair amount of growth. I put a photo of one of the plates that we got early on in this project. And you can see on this Petri dish, not only are there microbes there, but there's a lot of different kinds of microbes. They clearly have different phenotypes just on that solid media. So that was pretty encouraging. And that was really where we started, was, can we grow things? Yeah, go ahead.
So I'm fairly familiar with organisms that can tolerate different pHs. Acidophiles like low pHs, alkalinophiles have an issue with, or are able to like high pHs. Mm-hmm. What's interesting to me is that's always the concentration of hydrogen ions. And so the limit I'm aware of for microbial life is about 9.5. And so I'm curious if the pH has decreased on this concrete over time, or if any of your isolates tend to be alkalinophiles of a type.
Yeah, so excellent. Those are two excellent questions. Concrete does kind of mellow over time, right? Especially the surface of concrete. And in this initial study, we were really just taking things off the surface. So the surface of concrete doesn't have that crazy high pH anymore. We haven't isolated anything that requires a high pH to grow yet. Mm-hmm. We haven't tried very hard, because if you increase the pH above about 9, it's really hard, as I'm sure you know, to solidify the media. And so we would really have to do this probably in an all liquid system, which we can do, but we haven't tried it yet. That being said, and I'm giving away a little bit of the punchline here, in older concrete, we do see evidence of alkalophiles. So they are in there, but we don't have any in culture at the moment.
Well, so many things won't grow in the laboratory
right.
for a variety of reasons. And so
right.
when I'm hearing this, I'm thinking about the actual environment mineralogically.
Yes.
And we all know from geomicrobiology, there's a lot of things that take place. So I'm very excited to hear where this is going.
Oh, so excited. So, and when I started this, I really was thinking about concrete as if it were a rock, right? When I was in graduate school and when I was a postdoc, I really hung out with a lot of geologists. And that was sort of my internal bias, was this is a hard material, so it's like a rock. And that actually really misled me in a few ways. It is more porous than most kinds of rock. So I think there can be a little bit more transport inside than you would typically get inside a rock. But the other thing is that once we knew that there were microbes in concrete, we had some things growing in the lab, we decided, as you say, because not everything is culturable, to take a DNA-based approach, right? A cultivation-free approach. And so I was feeling pretty good about my abilities as a molecular biologist to get DNA out of whatever. And I tell you, it took us 2.5 years to reliably get DNA out of concrete. It was a real challenge. And part of that was because I said, they can get DNA out of rocks. We'll use those protocols. And none of those protocols worked for us. And at some point I had my student grow up something like a liter of E. coli and pellet them. So we got this huge E. coli pellet. And then I had her mix that with a big chunk of concrete that we smashed up to see how much of that DNA she could get back. And she got nothing back. And it turns out that the concrete matrix itself was binding the DNA and preventing us from getting the DNA out because it's so high in calcium.
Yes.
And there is also magnesium, and those divalent cations will hold on to the DNA. So that was our real problem. And so what we ended up having to treat concrete was like bone. The method that eventually worked for us was a method we adapted from a group that was getting DNA out of ancient bones, which is also a hard and dry material that's very high in calcium. But yeah, so that bringing up geology is fun because, that is also how I think too, right? Geomicrobiology. But it can be dangerous to have those kind of internal biases. So yeah, once we got DNA out of concrete, what we found in relatively young concrete, I was thinking, as you were thinking, right, that it must be full of extremophiles, right? To withstand this really crazy environment, they must be adapted to high salt, they must be adapted to zero water, they must be adapted to low nutrients, and this crazy high pH. But I really thought that the high pH was going to be the biggest selective pressure, and that we were going to see alkalophiles. And we did not see anything like that. Nothing. And it turns out, at the same time, we also did, this was a 16S study, our initial study. And at the same time, we also extracted DNA from all of the components, right? So the gravel, the sand, the cement powder that went into the concrete mix. And none of the things on any of those materials are extremophiles. And what you end up with in concrete is a very reduced microbial community that's made up of the things that came in with those materials. And so since there's no extremophiles in those starting materials, there aren't any in the concrete, at least when it's young. And
Julie, Julie, I was going to say, I say this to students and they're never happy about it, but boy, does this prove what I'm saying. Microbes don't care what you assume or think.
No.
They just don't.
No.
They just are, right?
Yes. This project has surprised me in so many ways. We've learned so much from it.
No, so I'm so pleased to hear that because so many times, and gently put, very famous scientists with many additional honorary degrees and once or twice a Nobel Prize will make assumptions about things that are completely unfounded.
Yeah.
Now, I'm not claiming I'm smarter than they are. Good Lord, I'm not. But what I am saying is that you have to have a microbial perspective, which is not ours.
Right. And you have to pay attention to what the data is telling you, right? And the data was telling us that concrete doesn't start out with extreme microbes in it, right? Although it changes over time. And again, if you think about the things that you see in and on concrete, they must be, in order to really thrive in that environment, they must be adapted, right? You can't just have your average bacillus hanging out in there doing a lot. Maybe you can. So in young concrete, we really saw microbes that were adapted to very low nutrient conditions. We saw a lot of oligotrophs in there. I was expecting to see, speaking of bacillus, I was expecting to see a lot of spore formers. And we do see a fair number of firmicutes and also actinobacteria, which don't really form the same kind of endospores, but a lot of them can form a dormant cell type that can sort of hold on and wait for a long time. So the firmicutes and the actinobacteria are a big fraction of the microbial population in that young concrete, but it's not the only thing by any means. But then we were curious about, what happens as concrete gets older. So a PhD student in my lab, Anders Killedal, went out with personnel from the Delaware and New Jersey Departments of Transportation and collected samples from roads, bridges, foundations around Delaware and New Jersey and extracted DNA from those samples. And we did metagenomic sequencing on those. We refined our DNA extraction protocol so we could get enough that it could be directly sequenced. And that was when we started seeing really cool stuff. We had specifically requested that they show us roads and bridges that had been damaged. So we wanted to get a spectrum of damage from totally fine to kind of old but fine to really old and pretty damaged. You can see a picture in the folder of a concrete bridge that there's a gel coming out. And this bridge they had diagnosed with alkali silica reaction, which is a pretty common mechanism of concrete damage in the Northeast. So what we found when we analyzed those samples was as concrete gets older and as it gets more damaged, we do see more extremophiles in that concrete. And we started seeing especially a huge signal from halophiles. So we saw a ton of haloarchaea. These are the kinds of microbes that you find in the Dead Sea or salt evaporator ponds. We also see a number of really well-characterized halophilic bacteria. We see a few alkalophilic bacteria, a few haloalkalophilic microbes from different species, but we really don't see them until the concrete is pretty old and also pretty damaged. So what we think is happening is that as concrete gets damaged, cracks open up and water can come into those cracks. That water will bring with it nutrients. So it will relieve the nutrient stress that the microbes in young concrete are feeling, right? Because it will bring organic carbon in, it will bring reduced compounds in. Concrete is so oxidized, there's almost nothing for them to use for energy in there. So this water, as it comes in, can relieve that stress. But when it gets into the concrete, it also will solubilize all the salts and alkalis that are in there. So you have this super high salt solution with nutrients in it. And the only microbes that would be able to access those nutrients are the ones that are able to withstand that really high osmotic stress. The other thing that we think is that water must be bringing microbes with it because those halophiles aren't in there early on, right? And so our next question and what we've been working on most recently was, where are those halophiles coming from? If they're not in the gravel that you get from the quarries in Pennsylvania, which we're pretty sure they're not, where are they coming from? Because here on the East Coast, we're pretty far from any of those huge, like super high salt environments. But we do also salt our roads all winter long. And so it was just a guess for a while that was potentially a source of halophiles, but we just got back a bunch of metagenomes from road salt. And the overlap with what we see in concrete is pretty big. It's almost entirely haloarchaea. So we think our hypothesis now is that road salt is actually a source of the halophiles that at least that goes into structures like roads. You can also see, there's a number of different places where you can see with your eyeballs salt accumulating on and around concrete structures. So a couple of the other pictures that I put in here are things like high salt stalactites coming down from the roof of a concrete bridge. Yeah. I've seen these a lot in this area around Syracuse. And also salt accumulating between bricks on the sidewalk. So we heavily salt the roads and sidewalks, like I said before. And that picture of like salt crusts in between bricks was a picture we took in June. The last time we salted the sidewalks here was probably early April. But there was so much salt put down that it forms these crusts in between the bricks, and we could still access that super high salt environment.
So it's a giant enrichment culture.
It is. It's a huge natural enrichment culture. And so what we've started doing just this past summer is trying to isolate some of those halophiles now that we know that they're there. So another picture that I put in the folder is a picture of 3 isolates that one of my summer students got. They're growing on 20% salt media. It's 200 grams per liter. And you can see, for comparison, the ocean is 3.5% salt. So this is much saltier than the ocean. And you can see 3 different kinds of microbes growing there. We're hoping to identify them in the next few weeks. But one of the surprises for me in this project was that the extremophiles that we did find, and it took us more than 10 years to find them, weren't alkalophiles. They're primarily halophiles. There is some overlap between the response to high salt and the response to high pH, right? These are both really strong osmotic pressures. But it does seem that there's more halophiles in there than alkalophiles. It may be a function of what's coming in, right? There's not a lot of there's potentially not as many sources of alkylophiles that would just wash into concrete
Hmm.
from what we normally do on roads and bridges. But
Well,
yeah,
for listeners and viewers, this idea of how cosmopolitan microbes are is a big sticking point, because a lot of times ecologists want to put an ecological view based on on mega organisms instead of
right,
microorganisms
right.
to this. So there was an ecologist named Bass Becking who had
Oh,
this
yes.
idea that microbes, all different kinds, were everywhere and the
Mhm,
environment selects. I know of people that base their entire laboratory course in microbiology from
mhm.
a gram of soil.
Right.
So this idea that everything is everywhere and the environment selects is one that is a viewpoint that may have some validity some of the time. But once again, I think even in this case, we put our own biases on it, I think,
Right.
honestly.
Right. I mean, if everything were really everywhere, I would expect to see more alkylophiles. I might
Yeah.
expect to see more xerophiles, things like you see in the desert. We see a little bit of overlap between, microbes in concrete and microbes in desert systems. We do see some alkylophiles, but we see this overwhelming signal from halophiles, and I really do think that reflects this very specific
Hmm.
input. One of the things I would really like to do is analyze concrete from regions where they don't salt the roads, right? Do we see in concrete that is of comparable age, 30 years, 50 years, 80 years, do we see halophiles thriving, or do we not because the inputs are different? Because I do think, yeah, go ahead.
Do you think that the road salt that you purchase has already been essentially spiked by these halophiles?
Yeah, so I think, okay, so full disclosure first, which is I know absolutely nothing about road salt. This is a very new part of this project, and I really haven't looked into it much. Um, but yes, I do think, there is overlap between what you see in road salt and what you see in salt mines, right? And
Okay,
so I
yeah.
think they're there in the mineral, going back to geomicrobiology. I think they're there in and on the minerals. And then I think they're selected long term. They're stored in these huge piles um, regionally. So yes, I do think that there's an initial selection, and that's what you end up with, a highly, highly enriched community in the road salt um, early on. And that's what gets washed into structures that we
I,
use.
I want to
For
make
road
you
salt.
laugh a little bit if I can.
Yes, please.
So there's an adult beverage called tequila.
Yes.
Right. Many bad decisions are involved with tequila. And you can sometimes see shot glasses made of halite.
Okay.
Now that is to say made of salt, and they tend
Yeah.
to be pink. And
Okay.
I have personally isolated halophilic organisms
No
from that.
kidding.
Yeah. So anytime that you see that reddish color, it's
Hmm.
normally it's normally rhodopsin,
Right.
bacteria rhodopsin, which is interesting to me. And by the way, just in case that you're feeling that I'm clever, I once put one of those shot glasses through my dishwasher. Yeah. Where'd it go? Right, exactly. So this idea, I mean, and I want for listeners and viewers, when you are dealing with something that's hypersaline like this, that's an incredible stress on cells because
Yeah,
they have to balance osmotically the solute concentration, which means those cells are driven to get rid of most of their water inside the cell to balance solutes. And this is why it's such an unusual community. And I'm going to be putting a couple of links in the show notes about halophilic organisms.
that's great.
I'll stay away from tequila shots.
Yeah, no, this has been really cool. And we do see evidence in the metagenomes as well of different strategies for managing that osmotic stress. So we see lots of ion transporters, so proteins whose job it is to move different ions in and out of the cell to help maintain that balance. We also see genes that encode production of compatible solutes, which are small organic molecules that are charged that the cells can kind of accumulate to really high concentrations inside the cell to help balance charges across the cell membrane. We also one of the things I have wondered for a long time is, what is the organic carbon in concrete, right? What are they eating? And we do see evidence that those compatible solutes are transported in and out of the cells. And so that's one potential mechanism of nutrient exchange. If the microbes are close enough to each other, they could be feeding each other. And that's one of the things they could be feeding each other. The other thing that I've been, related to this, the other thing that they could potentially be eating, there's a couple of things that I think are super cool. One is we do add polymers to concrete sometimes to make it easier to mix and pour so that it doesn't have a structural role typically, but it does make it easier to work with. But once the concrete is cured, it doesn't really have a job, but it is still in there. And that's a potential source of either organic carbon or electrons for the microbes in there. And a few years ago, basically just for fun, I picked a random set of 7 of our isolates. We have about 100 isolates in culture now from concrete. And I picked a random set of 7 of them. I think it was just whatever was alive in the lab at that moment. And tested them to see if they could grow on one of those polymers. And 5 of them could. So they can degrade those polymers, and it is not just potentially a source of organic carbon, but realistically, it could be a source of organic carbon for the microbes that live in there. Another possible source that I really don't want to work with because I think it's pretty nasty as a material is fly ash, but we often add fly ash to concrete to replace some of the cement powder. And that has complex organic carbon in it. It's really recalcitrant organic carbon, but it is organic carbon. And so if that's in there, it could potentially serve as a source of both carbon and, again, electrons. So we've been thinking about what are the potential sources of organic carbon. Another one is, I said at the beginning that the concrete microbiome is a very small subset of what comes in with the materials. And most things really do just die off, right? As concrete sets and cures, it heats up. A lot of things die when it heats up. And then once that material is established where it's hard and dry and salty, they just can't survive. Right? So they all die, but they don't come out. Those little microbial bodies stay in there, and that's another potential source of nutrients for whatever is surviving. So for a long time, I kind of thought of concrete as a real desert, and it is a super low biomass system. Don't get me wrong. This is not like soil, but it's also not as bad as you might expect, right? There are some sources of carbon. There are some sources of reduced nitrogen. There are some opportunities for the microbes that are adapted to that kind of environment. It's a place where there's going to be very little competition. And so we do see a small but fairly diverse community of microbes that survives in there.
So I wanted to tell listeners and viewers that, and again, I'm not trying to second guess microbial psychology, for lack of a better term. But here's the deal with oligotrophs is that a lot of microbes are just amazing at eking out a living on extremely small levels of nutrients.
Right.
As you're just alluding. So the corpses, as it were, of these of the organisms that were first in the concrete definitely could be used. Heck, there are organisms that can grow on the impurities in agar in the laboratory. Yeah,
sure.
and so you say, oh, yeah, but I want listeners and viewers to understand that that's one, when people, this is, if you think back to that horrific arsenic DNA story, this is a great example of that kind of thing, where people made assumptions about the microbes because in that specific case, they weren't really talking to microbiologists as they should have. But the power of what you've got here, you've got genomes from these organisms, you've got metagenomes, so you can begin to tease out the kinds of relationships they have, even in that low biomass community. I can't think of the name of the organism, but I'll put a link to it in the show notes, in a deep subsurface lithoautotrophic community. It's just it's a very small community, but it's able to eke out a living off abiogenic production of carbon dioxide and hydrogen in basalts.
Life is so wild. It's just so wild. You have these oligotrophs. Nobody wants to live in concrete, right? These oligotrophs are living on gravel, they're living in sand, they're living in water. They get dumped into a cement mixer. The whole thing gets mixed up. You make this big material. Every batch of concrete is brand new. So again, thinking about geomicrobiology, we think about change of environments on geologic timescales, right? So it takes a long time to develop a hydrothermal vent. It takes a long time to develop a desert. And the microbes have that much time to adapt to those conditions, right? Typically, evolution occurs on pretty long timescales. But every batch of concrete is new, and it doesn't typically include microbes that are already adapted to this really weird, hard, dry, salty material. And so every microbe that gets roped in has to either adapt fairly quickly or be something that can hold on just on the edge for a long time, or they die, right? And so we do see this like mass die-off early on, and then survival long-term of these very, low nutrient adapted oligotrophs. And the only things that we see in really big numbers in older concrete are things that come in later that are adapted to that kind of environment already. Does that make sense? But I think that concrete is super interesting because it is potentially an environment where we can see adaptation and potentially evolution on human timescales, right? Because every time it's poured, we're starting that experiment again.
From what you're saying, I'm going to call this a simple-minded question. If in fact you have communities that develop in the concrete and you have isolated some of them, has anyone tried to put those microbes into freshly poured concrete to see if it develops differently?
Oh, this is actually an experiment that we're starting to do with a colleague up at Clarkson University. So I have a collaborator, Robert Thomas at Clarkson, who's a concrete materials guy. And we're not starting with rebuilding the community. I'll come back to that. What we're starting with is how many microbes can concrete tolerate?
Oh, okay, that's
because
good. Yeah.
This is such a low biomass system, but if people are thinking about putting microbes into concrete for biorepair, which again is not what I'm working on, they're experts in that field. But for biorepair or for any other purpose, we have to know how many you can put in there, right? Before you lose the concrete structural properties, because we don't want to lose performance, right? We use concrete for structural reasons, right? It does a job and we don't want to break that job like when we give it a new one. So that's what Robert's doing right now, him and his student. I have been thinking about like synthetic communities related to concrete, but I feel like I don't yet have enough data because concrete is a really local industry. So DOTs or construction companies, they source the materials for concrete locally. Nobody ships gravel across the country to make sidewalks, right? So you get your gravel locally, you get your sand locally. The water is supposed to be potable water, but it's often just the local, fire hydrant or whatever. So all of this is local, and what we don't know is, are the concrete microbiomes also local, right? Because they reflect the microbes that are on those starting materials. Do you end up with different microbes in every region? Or because you always end up with the same material, with the same conditions inside it, is there a really strong selective pressure that means that you end up with the same small microbial community every time, regardless of what you start with? And we really just don't know the answer to that question. Yes, this is one of the things I would really like to do, in the near future. But I haven't found a way to fund travel around the world to bring concrete home yet.
Well, no matter how you would approach the problem, you'd have difficulties that occurred. So, for example, if you said, I'm going to pour a brick of concrete to use as my model system, you don't know that really reflects the way concrete exists in a road or a bridge.
Absolutely.
That's the thing.
Yeah. No, absolutely. And this was, so the initial study that we did was actually exactly this. We poured a whole bunch of little cylinders, approximately brick-sized, and we wanted to see what would happen as that concrete naturally weathers. So we put them out on a green roof because we wanted them to be exposed to normal environmental conditions, but we wanted to avoid input from like foot traffic or dog poop or whatever. You knew dogs would have to make an appearance. Yeah.
Seagulls, that's the issue, right?
Well, the seagulls you can't do. This was a roof, so the birds was still possible. And we did see small changes over the course of 2 years of weathering, but not very big ones. But I do think now that a lot of the changes in concrete are probably driven by environmental inputs rather than change of that population, of the internal population over time, right? And so it's possible that if we were to do the same experiment, but instead of pouring small cylinders, just take a big piece of concrete. There are some big projects on the east and west coasts and in Texas looking at concrete weathering over decades, right? And so potentially taking samples from some of those would be really cool, right? Where they would have more normal environmental inputs. But I don't know.
Karen Lloyd, who's a
Mhm.
previous guest on this podcast, and colleagues came up with a concept, and it might be controversial, and I may be mispronouncing it, but ionophiles,
Okay.
the idea of very, very slow growing organisms.
Mhm.
Okay, that in that context prefer it. And then when we talk about an oligotroph, this is really important because most of our focus is something that I call coli centricity. We
Mhm.
tend to think that microbes are like E. coli, which is a copiotroph and
Right.
grows relatively rapidly, that kind of thing. But most microbes aren't like that. So when I hear about this kind of insular environment where if growth is occurring, it's occurring slowly, it made me think about ionophiles for that reason.
Yeah, I agree. Most of the things that we have in culture, and I think this is just a bias from culturing, right, are
Mm-hmm.
things that grow fairly quickly. I will also say that post tenure, I feel like I can afford to let things to wait a little longer. And so we do now have some other ones that grow more slowly, right?
Sure.
But yes, I agree. There probably is this really big population of things inside concrete that grows so slowly that it's hard to observe. One of the things that I would really like to do is something like an activity assay, fluorescence-based activity assays. And we have done a little bit of confocal microscopy with chunks of concrete that we doped in a culture. And just because I wasn't sure if there would be enough fluorescence from the native population to see it without doping. So we started with a doped culture, and we can tell the difference between cells and concrete. We haven't followed up with activity assays, but because we know we can tell the difference, that is a possibility now. And I am very curious because I don't know how active they are in concrete. I know they're not dead, right? But there's a difference between not dead, holding on by their fingernails, and really growing. So
I don't
again,
know.
something we really don't know yet, but we're thinking about testing.
I love the idea of applying a human-centric metaphor. So if holding on by our fingernails is something we're used to, what would be the equivalent for microbes? Holding on by the pili or flagella? What would you say? I mean,
Yeah,
that's a
probably
really
the
interesting
pili.
thought. Yeah. Because in fact it is, right?
Right.
They're really good at that. So this is, and I want to emphasize when we talk about this, folks like you and I are going to get nerded out by the environment and all the rest, but there's real importance to this research as you begin to understand the factors that control how quickly this concrete degrades.
Yeah.
I put to one side the idea that I've read about before about biorepair of concrete, but we should understand how to deal with concrete that's breaking down and minimize that, because as bridges, they're going to start to break down from this kind of thing over time.
Yeah, I'm not sure how much damage they're doing. And the reason I'm
Oh,
a little
okay.
skeptical about the damage is that the biomass is so low.
Okay.
And
That's
so
good.
on the scale of a bridge, I'm not really worried about it. That being said, I don't think that they can initiate damage. I don't think there's enough of them. But I do think that they can potentially exacerbate damage, right? And I am concerned that two things are going to potentially kind of inoculate our concrete with things that can thrive in there and can potentially contribute to a negative feedback loop. And one is the way that we salt our roads. This is potentially an issue for a lot of environmental reasons already, right? We end up with salinization of local waterways and local soils. And of course, we want people to be safe when they're driving. So I understand that there are competing concerns here. But I do think that road salt application does have a number of environmental concerns associated with it. And one of them is it's bringing these microbes to our infrastructure that can potentially degrade the infrastructure. The other is sea level rise, right? As we have salinization of either groundwater resources or we have flooding from overtopping of salty water, in coastal areas, we're also bringing in microbes that are adapted to relatively high salt conditions, and we're bringing them to this concrete infrastructure, and they do have the potential there to grow and to exacerbate the damage. If something is adapted to that environment and is able to thrive there, then the biomass will increase, and then you have greater potential for damage. So in general, I'm not worried that microbes are going to make our bridges fall down, right? But I do think we need to be aware of the possibility that we're bringing microbes to that infrastructure that could potentially have long-term consequences.
In the worst case scenario, Julie, if you consider how damaged concrete, I'm wondering, in that worst case scenario, is there a microbial involvement in the further breakdown? In other words, is it accelerating after you get a certain degree of damage?
I think that that's possible. So as you know, microbial metabolism generates some degree of acid, right? Just from normal energy metabolism, that we build up this proton gradient across the cell membrane. And then a lot of microbes also produce organic acids in different conditions. And so that acid production, I think, is probably has the most potential to do damage in concrete. But again, because the microbial biomass in concrete is so small, I think that the damage would likely be localized. I think the possibility for further damage starts coming in when you have extensive colonization. And that's most likely on the outside of concrete. And this has actually been really well characterized in places like sewage systems, where you have those huge concrete sewers, right?
Yeah.
And you have thick biofilms that accumulate, you have sulfate in the water, you get sulfate reduction, and you have production of hydrogen sulfide that volatilizes, condenses on the roof of the concrete sewer, generates sulfuric acid, and then you have pitting on the roof. It's called crown corrosion. So in places where you have a lot of biomass, this definitely has been documented. But I do think that in most concrete where you don't have a consistent supply of water and a consistent supply of organic carbon, which you do have in sewage systems, that type of damage is going to be much smaller if it happens. It does have the potential to be happening, but at smaller scales.
Part of this sounded to me almost like tooth decay. Except it's not, because tooth decay is much more damaging and immediate by comparison.
Right. Although if you have failure of a 6-foot sewage main, that's a real problem. So,
I agree.
But yes, and like I said earlier, concrete was surprisingly more like bone than like rocks. So I do see some parallels there. There are also a number of people in sort of the synthetic biology space who are thinking about using microbes in concrete to do jobs, right? So there's a company in North Carolina that's using microbes to make concrete bricks. There are a number of other research groups around the country that are interested in using microbes for biorepair, for bioproduction. And I do think that there's potential there for putting microbes into concrete to make it either more sustainable or to do a better job, right? Or to do different things. We can add functions to concrete potentially. And I do think that there's a lot of space for creativity there as well. So I think this is a pretty exciting field. And from a basic science perspective, because the concrete microbial community is pretty small, it is a relatively straightforward community to analyze, right? You can get a complete metagenome. You can assemble really high quality individual genomes from a metagenome with not that much sequencing. And so what that means is that it's a pretty good model system for microbial community assembly, right? Mm-hmm. And potential interactions, because it's relatively simple. And so we can use this system as a model microbial system for understanding what are the most basic interactions that occur in mixed microbial communities.
Do you find autotrophs in this community?
We do find a few, not a lot, but we do have a couple of metagenome assembled genomes, MAGs, that indicate autotrophy. We have a serpentinomonas mag in one of our samples. So we do also see in surface exposed samples a number of cyanobacteria, even alkalophilic and halophilic cyanobacteria. So we do see evidence of autotrophy in these older samples. I'm not sure where those came from. I didn't see any evidence of those in the road salt data, but we do see them in this older concrete. And so, yes, there's potential for autotrophy. We see some evidence for fermentation. So there's potential for generation of hydrogen, which is a super useful electron donor. So that would help to relieve the energy stress that I think a lot of these microbes are feeling. So we have circumstantial evidence for the possibility of nutrient exchange. I won't say it's real solid because I really don't know where these microbes are in relation to each other, how far apart they are, right? When you have a 10-gram sample, it's really big. And this distance for a microbe might as well be 6,000 miles. It's so far. And when it's this distance in a solid matrix, it's even further, right? The possibility of nutrient exchange from here to here, in the absence of things like cracks, is just almost impossible. So we don't really know how feasible nutrient exchange is, but we do know that the metabolic possibilities are there.
You know, Julie, there in the old now movie Jurassic Park, when the ever quirky Jeff Goldblum says life finds a way,
Always.
I want to put microbial in front of that.
For sure.
Because that's what I'm surprised by in so many ways. Look at cable bacteria that can essentially create a snorkel to dump their electrons.
It's so wild.
And that's the equivalent of, if not 6,000 miles, hundreds and hundreds of miles.
Oh, for sure. For sure.
Right. And I'm not saying that's taking place there. What I am saying is that within this simple, well, relatively simple community that you've chipped out. I was going to say uncovered, but chipped out's better.
It is.
Now, this is a wonderful way to look at it and start to think of it that has relevance to the built environment in general. You can answer so many interesting questions with it, and you're really to be commended for this.
This has been such a fun project, and it really does tend to capture students' imaginations. So students get pretty excited about it. I presented 2 slides about this project to a group of sophomores earlier this week, and I got 15 minutes of questions.
Yeah.
They were so excited. They had all these ideas. Like it really just gives them something to where they can kind of apply scientific imagination. And because we know so little, their ideas are completely reasonable. So it has been a really fun project for a lot of reasons, but that's one of them.
Oh, I really like this idea. And I mean, finding something that everybody encounters every day and putting that, I need to have a microbially colored set of glasses. I don't know what that would be. But that's the goal, is to make you look through a set of microbial glasses at things that you see every day.
Yes. This is actually something I've been doing in my classes for almost 10 years now, is at some point during the semester, we do a microbe walk. And we just walk around campus, and I point at microbes that you can see with your eyeballs. So lichens always show up. Concrete always shows up. The soil, of course, always shows up. I always make them smell it.
Yeah, that's fun.
Yeah, there's a number of different spots on campus, and I can usually find them on different campuses too, where you can point at things and you can't always see them, but there's usually some evidence that you can see or smell or experience in some way.
I think you should make a video of this and share it, because I think the idea of a microbe walk would be wonderful. And you're going to say, well, you can do that anywhere, and you can, right? But what a wonderful idea to share how important microbes are.
Yeah, I've been thinking about doing that. Maybe I will.
Yeah.
But it's always fun. And it just gets students really engaged. You know, I usually send them on a scavenger hunt afterward. That's the assignment, right? Is to send me a picture of a microbe that they can see.
Yes. What a wonderful idea. So are you familiar with the journal GEMBI?
Yes, I talked to them last summer at ASM at Microbe.
Yeah. You should put this together as a classroom activity, because it's wonderful.
Yeah. It's been a lot of fun. So
yeah.
This is really,
go ahead.
I was going to say, this is really how I decide what to do in my classes and in my research, which is it fun for me? So
sure. I mean, this is because, honestly, with all due respect, we're not in it for the money, are we?
No.
I may be misremembering this, but my understanding was making of concrete is one of the biggest sources of CO2 production.
The reason it has this really high carbon footprint is the making of the Portland cement. So to
Okay.
make the Portland cement, we take lime and we fire it to something like 2500 C. And so the energy input for that process is what gives it this enormous carbon footprint. And so among the ways that people are thinking about to make concrete greener is to replace the Portland cement with different things. So sometimes we replace it with things like fly ash, which is a waste product otherwise. And that's often done in the Northeast because it does also, in addition to replacing some of the cement powder, which is expensive and has this huge carbon footprint, it helps to mitigate against certain types of chemical damage, like alkali silica reaction. There are some alternative cements that people are now evaluating as well, that either reduce the amount of Portland cement that you need to use or replace it. And that's one of the things that I'm doing in collaboration with faculty at Clarkson University is, are the microbiomes of those concretes different? We don't have any of that data at the moment.
Yeah.
So I can't say anything I can say that we're working on it, but we don't know.
Many types of concrete are reinforced with rebar, iron, steel, and that might certainly give some change to the environment locally because of the availability, say, of iron.
Yes.
Am I misthinking this?
No, it's totally true. And those metals can also provide some of those electrons that I was saying were hard to find in concrete, which is so oxidized. So yes, it does change the system. And there is pretty well documented microbial corrosion of rebar in concrete as well. I have stayed away from that because we were interested in what was in and on the concrete matrix. And we thought that rebar, I think we were right about this. We thought that the rebar would make it more complicated. So we stayed away from those kinds of samples to the degree that we could. I think there was rebar in some of the field samples that we collected in Delaware and New Jersey. Although I don't know if I have that information.
You do have to pick and choose the system carefully, obviously. And,
yes.
and one of the things that you mentioned is the limited population diversity and density. And that can be an advantage once you, as you did, can work out ways to elaborate it and study it.
Yeah.
So I appreciate this very much. Another question that occurs to me is about Roman concrete, because everyone's read about Roman roads that seemingly last forever. Yeah. And is it kind of what you're talking about related to this, or is there perhaps there's less damage from Roman roads for some kind of characteristics? Yeah. So yes, I get this question all the time. The short answer is I have absolutely no idea. The longer answer, and also I really don't think that the Italian government would like it if I went over and sampled with a drill. But every 3 years or so, I see a new article about how we've solved the question of Roman concrete. Why has it lasted so long, but our bridges are falling down after 30 years? What's the difference, right? And my impression, again, just my impression of the field is that we still are figuring out what was unique about Roman concrete. So among the things that I've heard is that they may have used seawater rather than fresh water, at least for some of their structures. I think they used volcanic ash rather than Portland cement that is manufactured. The large aggregate, the gravel that they used in that was also locally sourced and probably different. And I did see a really interesting article saying that the way that mineral interacted with water, it'll slightly dissolve and then recrystallize in a different, stronger form. And so in some cases, that Roman concrete may not have just been self-healing, but self-reinforcing, right? But it's really hard for me to say. I'm not really a concrete materials expert. I read these papers, and every 3 years, a different one comes out with a different, we figured out the secret of Roman concrete. So I'm not really sure. But I will also say that concrete is a really heterogeneous material in part because it is locally sourced. And I think that's been true for probably 2,000 years, right? You just can't move really big quantities of heavy material very far. So it does kind of, practically speaking, it has to be locally sourced. And so even if we know what
what made Roman concrete so long lasting, it might not be possible to recreate that in other places because it may depend on the local availability, the availability of specific local minerals. You can keep that answer or not as you like, but I really don't know.
No, I think it's lovely. And something I want to emphasize to you, and I mean this sincerely, is that this is 100% applicable to almost anybody, regardless of background. And that's what's so cool about today's subject.
Yeah, like I said, I really like this project for that reason too. Around the time that I started it, I spent 7 weeks in Indonesia on a different project doing field work. And, that was a great experience too. But a 7-week field campaign is just not practical ever, And for this project, I don't have to do that, right? I can go, okay, so the DOT doesn't like it if you do this, but I can go outside with a hammer and collect a sample, right? Or I can call the DOT and say, what do you have in your archive?
Right.
Or, actually, when my student went out with the New Jersey DOT, they said, we don't want you drilling into the structures. And he said, fine, how are we going to collect them? And they said, just wait. So they stood under a bridge, and every time cars drove over, little pieces would fall down, and then he would pick them up. Very quickly. So, the sampling is much more straightforward, and it really is a very easily accessed, but very extreme and very weird environment.
Julie, we had Arturo Casadevall on the program a couple of times, and I couldn't believe how seriously he presented this, and it just made me laugh so hard. They were showing where you would find fungi, and what they were using to pick up the fungi were starbursts.
Nice.
The candies.
So fungi.
It's in the paper that they use starbursts.
Overly honest methods. I miss that hashtag so much.
Yeah.
In the category of overly honest methods, the road salt that I extracted DNA from last January, I think it was, just came from the DOT, right? So it's filthy. One of my students went to the local salt storage facility and asked the guys there, hey, can you give me some road salt for science? And they were like, sure. So they gave him a bucket full of salt, but it's real dirty. And so to extract the DNA, we decided we would dissolve it first and then filter the cells, collect them on filters, and so treat it basically like a water sample. But the water was so dirty that I passed it through a coffee filter first to get rid of the literal dirt. It was kind of amazing. And I'm still deciding how I want to put that in the methods section.
To remove extraneous debris.
Right. Yes, it does say something like that. One of my postdocs years ago, again for a different project, passed water samples through a stocking, like a nylon stocking. And I think we called it 1 millimeter mesh in the paper.
Yeah. So I want to say I'm watching our time here,
okay.
and here's a secret that I think most people have figured out. I do this podcast as much for me as for anybody else. So I've gotten a lot out of it, and my guess is listeners and viewers will be intrigued as well. Do you have perhaps 3 take-home lessons from this presentation today?
Sure. Let's see. 1, there are microbes in concrete. I didn't believe it when I started, but there are. 2, some of them are real weird, and may even come from things like road salt that I also didn't think had microbes, but it turns out that it does. 3, you are not in any danger from the microbes in concrete.
I think that's beautiful. No, those are wonderful, punchy things. And this is the goal. Now, there are people who listen to this podcast who are, super skilled scientists, and that's cool. And then there are people who are students in microbiology, and that's wonderful. And then there are everyday people who maybe are interested in science but don't know that much about it. So this has been a real service today, and I want to thank you.
Well, I hope everybody enjoys it. It has been a lot of fun, not just to talk about it, but to do this science and to work with the students who get really engaged. So thanks, everyone, for your attention. I appreciate it. And Mark, thank you for this invitation. This was really fun.
I really appreciate your time. I wish you and yours, and tell your students hi.
Will do.
Are you teaching currently?
Yes.
How many students do you have?
Let's see. This semester I'm teaching applied biochemistry, so we have 15 students in that class. Next semester, I teach biochem 2, which is closer to 40.
So I'll send some extra stickers to you.
Oh, awesome. Our students will literally do anything for stickers.
Have you noticed? Yes. Thank you so much for your time, and I wish you and yours so well. Okay. Bye now.
Bye.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, as always with epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Julie Maresca can be found in the Department of Chemistry at the SUNY College of Environmental Science and Forestry in Syracuse, New York. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music that makes me smile every time I hear it. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.